Thank you very much. So, if you look back in the long history of RNA, it starts back in the 1960s when interferon was first described as being produced by RNAs, RNA of a variety of types. This was followed by a bunch of other seminal findings that led to ultimately therapeutic uses of RNA, include discovering the cap structures, poly A tails, abilities to make long RNAs, which previously had been impossible. And the first time an mRNA was actually injected into an animal and an encoded protein observed was in 1990. So what are the benefits of nucleoside, of a modified or any RNA therapy? And there's a number. The first and probably the most critical compared to other nucleic acids is that it only needs to be taken up into the cytosol to be translated and have efficient function. The next are a series of safety findings. There's no possibility of genomic integration. There is no, it's a simple coding sequence with UTRs and poly A tails. There's no replication. Its translation is limited, and that's defined by the length and sequence of the RNA and poly A tail. With the introduction of nucleoside modifications into RNA that Katie Carrico and I described, it's gotten rid of the immunogenicity of that RNA. And most importantly, when you think about the delivery of therapeutic proteins, proteins have to be made in vitro, in CHO or other cell types. With RNA, the host cells are making the protein. Other advantages of mRNA, it's highly translatable and has very high transfection efficiencies, greater than 80 to 90% in primary cells and in vivo. It's very easy to make. It's a single reaction to make RNA, a single reaction to purify, and it's very cost effective. So what are these advantages of mRNA? So if you think about a pharmaceutical company making a therapeutic protein, this is what their GMP facility looks like. They have 50,000 liter vats where they have their CHO cells or whatever other cell type they use to make the protein. That protein isn't secreted into media. They then have to undergo a series of purifications to get the protein away from the media and to purify it. These purifications use multiple columns of various types. The risk that you run into is that when you purify these proteins, what if you misfold a protein? Or what if you modify a glycosylation site, or you misglycosylate? And there have been a number of examples of this. Probably the most telling was one about 10 years ago when a pharmaceutical company changed how it formulated erythropoietin that was used as a therapeutic. That led to a number of deaths due to the introduction of immunogenicity into that EPO. So the patients that received it made antibodies against the delivered EPO protein that cross-reacted with their own EPO, leading to aplastic anemia. With mRNA, you inject the RNA, the host cells make the protein. There's no chances of formulation or production leading to aberrantly produced proteins. So I showed you that RNA was first injected into an animal in 1990, and it essentially went nowhere after that. And the question is why? And this is the summary reason of why. At present, there have been 17 different innate immune sensors. These are host sensors that recognize RNA. They do a variety of things. They produce type 1 interferons. They produce pro-inflammatory cytokines. They induce apoptosis of cells. We characterize them by different categories. There are the cell surface molecules, endosomal, cytoplasmic, extracellular. In particular, these two, along with RNase L, when they're activated, they directly inhibit translation. So when Katie Carrico and I first started studying this, the first question we asked was, are all RNAs equally immunogenic? So what we did was fairly simple. We took host mammalian cells or bacteria, and we isolated the different types of RNA from these organisms. And then we analyzed each for immunogenicity. So the way we did this is we actually used monocyte-derived dendritic cells. And the reason we chose this cell type is that the dendritic cell has most of those 17 RNA sensors, not quite all, but almost all. That way we're able to look broadly at all of the sensor recognition of different types of RNA. And the data was very interesting. So the positive controls are poly-IC and R848. We added each of the RNAs with or without prior RNase treatment to show that it's an RNase effect. So in all of them, RNase got rid of the activity except R848, which is a chemical stimulant of Toll-7 and 8 in human cells. But what was interesting is in vitro transcribed. So this is 4 nucleotides only in an RNA of any sequence that you choose, was highly immunogenic. It produced a lot of TNF-alpha. Also produced type 1 interferons, IL-6, IL-12, and a variety of other cytokines and chemokines. But what was interesting is that bacterial RNA was highly immunogenic. Mammalian tRNAs were non-immunogenic, did not induce any cytokines from these cells. Mitochondrial RNA was immunogenic. The rest of the RNAs had varying levels of immunogenicity. So that made us think, and what we did is we plotted nucleoside modification as a function of immunogenicity. And what our hypothesis was is that it's modified nucleosides block the immunogenicity of an RNA. Our thinking was that that would allow a host to differentiate self-RNA from pathogen, bacterial or viral RNA that was introduced into a cell. And we got a very nice association. The lower amounts of modified nucleosides or number of modified nucleosides led to increasing levels of immunogenicity. As a recap of what nucleoside modification is, there's over 100 different types, probably upwards of 140 right now. They are all post-transcriptional, made after the RNA is made. They're introduced by a variety of enzymes using a variety of systems. Many are highly conserved. You find the same nucleoside modifications in bacterial ribosomal RNA as you find in mammalian ribosomal RNA. There's different types of modifications. So there's a pseudouridinylation, which is essentially flipping the axis of the ring. There's a number of different base modifications that occur both on the sugars and on the nucleosides. What's interesting is that many of these modifications don't change the ability of the nucleoside to mediate base pairing to its partner. So the introduction of a 5-methyl or 6-methyl doesn't alter the ability for the U and T or C and Gs to bind and recognize each other. What's also interesting, I mentioned this before, is that the number of pseudouridines is almost 9 times higher in mammals than it is in bacteria. So the next step is, let me just review how you make RNA and how you make modified RNA. In general, the procedure for the past 15 years has been to take a plasmid, put a promoter for a phage polymerase. In this case, we're looking at T7. After the promoter, you can put in a variety of 5' and 3' untranslated regions with a coding sequence in the middle. And the poly A tail can either be encoded in the plasmid or can be added enzymatically afterwards. You take that plasmid and linearize it at the end of either the poly A tail or the 5', 3' UTR, and then using a phage RNA polymerase along with the NTPs with or without cap analogs, and the enzyme makes copies of the DNA as mRNAs. Those mRNAs can be further modified. You can add a cap if one wasn't present in the transcription reaction. You can extend the poly A tail. The way we made nucleoside modified RNA is we essentially trick the polymerase, and instead of adding a UTP, we added a pseudouridine triphosphate, or a 5-methylcytidine instead of CTP. So we took RNAs where we changed every cytidine to a 5-methyl, every adenosine to a 6-methyl, every U to a pseudouridine or to a 2-thiouridine, and asked what did that do to the immunogenicity of the RNA. Here we're measuring two different cytokines, TNF-alpha and IL-12. This is 12 hours after addition. This is the same RNA, identical sequence, with different nucleoside modifications. And what we found is that the introduction of modified nucleosides greatly reduced the immunogenicity of the mRNA. So that answered a hypothesis that nucleoside modifications could reduce the immunogenicity of the RNA. We looked in primary human dendritic cells. Everybody complains about in vitro produced cell lines, and we got identical results. And it seemed to be primarily uridines that mediated the reduction in immunogenicity. So DC1s are myeloid in nature, DC2s are plasmacytoid in nature, and in both, the modified U's greatly reduced cytokine production, the immunogenicity of the RNA that was added. What was most striking was the next experiment that we did. And we were both surprised and didn't expect this to happen. But the modified nucleosides were not only were they translated, but they were translated better than the unmodified RNAs. We spent a bunch of years trying to figure this out, and what our conclusions came down to is that multiple receptors in the cell, RNA sensors, don't recognize modified nucleosides. And the increase in translation was in part due to a lack of interferon that upregulates many of those RNA innate immune sensors, and a lack of activation of PKR and oligoadenylate synthetase RNase L. Activation of either of those directly inhibits translation. So it's not that the modified RNAs are translated better, it's that the unmodified RNAs are translated worse because they activate RNase OAS and PKR that inhibits their translation. Another thing we found out during these studies is that the phage polymerases aren't perfect, and they introduced contaminants into the produced RNA. We set up a dot blot using a monoclonal antibody that recognizes stretches of double-stranded RNA. And we found that for any modified nucleoside or unmodified nucleoside containing RNA, there were measurable levels of double-stranded RNA contaminants. We developed an HPLC purification procedure that separated single-stranded and double-stranded RNA. And when we repeated the dot blot, the purification got rid of the dot blot staining. It got rid of the double-stranded RNA. When these RNAs were then tested in more sensitive dendritic cell assays, They produced no cytokines, no interferons, no IL-6, pro-inflammatory cytokines. We measured genes in these cells, and very few genes were upregulated with the purified modified RNA. So the question then comes, what effect does that have on translation? And again, we were surprised by the results. We could see that the combination of using modified nucleosides and the HPLC purification in red could increase translation by 4 logs. What was striking is that this was much more apparent in primary cells than in cell lines. That's likely due to cell lines lack significant expression of PKR, OAS, and many other RNA sensors. So we then asked, what happens when you put these RNAs in vivo? And the first assay, we used luciferase since we could image it. We would inject mice with luciferase encoding mRNA. We would follow that up with an injection of luciferin, which is the substrate for luciferase, and we would image them in a biosensor. What we found is that we were using lipid nanoparticles as our complexing agent. LNPs are known to bind ApoE and target the liver. So any systemic injection, IV, IP, gave high levels of liver translation for a short period of time. What was most surprising is that intratracheal administration also led to liver expression. What that means is that these LNPs have the ability to cross multiple basement membrane containing layers in order to reach the blood and ultimately reach the liver. What was also striking is that peripheral sites, like intradermal or subcutaneous, translation remained at the site of injection and continued on for many days. Here I'm showing 9 days. We've gone out to 2 weeks after intradermal injection and continue to get translation. This is just a direct calculation of amount of protein produced and duration. IV and IT, both of which target the liver, were very short. Intradermal and subcutaneous, we had continued translation for quite a long time. So the next question that comes, well, what can you do with RNA? What kind of therapeutics can you think about for using with RNA? And a number have been used. These involve both cancer and infectious disease vaccines, delivery of CARs, chimeric antigen receptors, introduction of multiple protein replacements, here, introduction of monoclonal antibodies, delivery of gene editing technology, delivery of transcription factors. It's basically whatever you can think about delivering, you can likely do with RNA. So these are what's been published, or much of what's been published so far. And I'll take you through a few of these just to show you what the efficiency of RNA delivery is for these different types of proteins. The biggest limitation to delivering RNA is the delivery systems. The LNPs, which are in phase 3 clinical trials and are safe, deliver to the liver unless you inject at a peripheral site. Other delivery systems involve direct application, so intratracheal administration to get to the lungs, or painting on the skin to target the skin. There aren't good delivery systems developed yet for targeting other specific organs. Endothelial cells have been described. Most other organs have not been described. So the very first study that used mRNA, and this was the only RNA study, therapeutic study that occurred prior to Katie and I developing nucleoside modified RNA. The authors injected vasopressin, ADH encoding mRNA, into the hypothalamus of a rat that was known to be deficient in vasopressin. ADH vasopressin allows the kidneys to concentrate urine. So in its absence, urine is the same osmolarity as blood. In these animals that were injected either with saline or nothing, they're unable to concentrate their urine. But the injection of 50 nanograms of mRNA lacking a cap and lacking a poly-A tail that encoded vasopressin led to a correction in the ability to concentrate urine. They also injected 5 micrograms of RNA from hypothalamic cells. This was the first demonstration that RNA could be used therapeutically to deliver an absent protein. Other studies have been done. A number of groups have studied erythropoietin as a therapeutic delivery system. We chose it because it was easy. It's easy to measure its effect, and it's easy to move up in animal species. The first thing we did is we made a number of different erythropoietins, either nucleoside modified or unmodified, or encoding another protein, and injected these into mice and followed the EPO levels over time. What was striking is that 0.1 micrograms led to enormous levels. This is nanograms of EPO measurable for 5 days. EPO has a very short half-life in vivo. The protein, the purple is a protein dose that's about equivalent to what we give a human. So we're over 10 times greater than a human dose with 0.1 micrograms. We then did a dose response. Erythropoietin acts on the bone marrow to produce reticulocytes as a nucleated red blood cell that then go on to become standard red blood cells. And we found that as little as 10 nanograms of mRNA could stimulate a supraphysiologic response of reticulocytes. This is protein injection. This is 3 times the human dose. Unmodified EPO, we get a little bit of a bump in reticulocytes, about equal to 0.001 micrograms, 1 nanogram. So nucleoside modification is about 1,000 times better than an unmodified RNA. A single injection could increase the hematocrit in these mice up to very high levels for an extended duration of time. And most importantly, the RNA was non-immunogenic. We measured circulating cytokines after a single injection. Unmodified RNA induced very high levels of circulating type 1 interferon. Modification, we had nothing. Surfactant protein B has also been delivered. So surfactant protein B deficiency is a very rare disease that leads to the lack of formation of effective surfactant. It results in shortness of breath, respiratory insufficiency, and often death in most children afflicted with it. What surfactant does, it has 2 effects. It has a direct effect on surface tension, where it disrupts the cohesion of water molecules and allows a lack of surface tension, as well as an indirect effect that is similar. When you think about delivering surfactant protein B, though, you have to think about the other diseases of the lung that could be similarly treated, including things like cystic fibrosis or alpha-1 antitrypsin deficiency. So the authors delivered a nucleoside modified SPB encoding RNA by the intratracheal route, and most importantly, 2 injections during a single week increased the life expectancy, and continued injections of RNA twice a week greatly increased the survival of treated animals. If you look at the histology of the lung, this is normal lung. You have very thin septate for the alveoli. In the absence of SPB, the septate increase in size because of inflammation and the septate breakdown. Treatment with mRNA gave you essentially normal-looking septate. A second approach this group did is they tried delivering a zinc finger nuclease to correct SPB deficiency. What they essentially did is they put a new promoter in front of a doxycycline-sensitive SPB gene. They used zinc fingers delivered with modified nucleosides into the lung, and they were able to greatly extend the lifespan of animals with correction of the SPB deficiency. The reason the animals later on died is that they weren't targeting stem cells. So after the type 2 alveolar epithelial cells were replaced by host cells lacking SPB production, the animals later died. But again, what you see is that with RNA or AAV correction, you maintain the alveolar structure versus in its absence, you have destruction and inflammation throughout. The heart has also been targeted with nucleoside modified RNA. Now the difficulty here is that there's no way to target the heart through intravenous or other delivery systems. So these authors directly injected the RNA into the epithelial surface of the heart. What the model was is they introduced a myocardial infarction in the mice by clipping a cardiac artery, and then injected VEGF encoding RNA into the epicardial space. When they did this, they improved the survival of mice given a heart attack. They measured heart function by echocardiography. This is the important. A normal mouse has an ejection fraction of around 65%. In mice that were treated with the ligation, they had very poor ejection fractions and died. Mice treated with VEGF RNA mostly maintained their ejection fraction, so they improved their heart function with modified RNA, which led to a better lifespan. Therapeutic monoclonal antibodies can be delivered with mRNA. In this study, we chose a broadly neutralizing HIV monoclonal called VRC01. The way we did this is we made two RNAs encoding both the heavy chain and the light chain of the RNA, mixed them together in an LNP and injected them. We then followed VRC01 levels in mice. We could see that a single injection of 1 milligram per kilogram of RNA gave us levels in the 170 microgram per mil range, so incredibly high levels. We could also repeatedly deliver the RNA and maintain trough levels above 40 micrograms per mil. We did this in humanized mice. Here we're comparing a dose of 600 micrograms of VRC01 protein, which is the therapeutic dose for a mouse, versus 30, 15, or 7.5 micrograms of mRNA. That 15 micrograms was a little bit lower than the therapeutic dose. When we gave these to humanized mice and challenged them with HIV, we had complete protection at 15 micrograms, so 1/30th the amount of protein, and 1 out of 6 was protected at a lower dose. So, what other things can you do with RNA as a therapeutic? And here I've just spelled out what some others in the group are going to talk about later on, including delivery of tumor-associated antigens as cancer vaccines, CAR receptors, and a slew of pathogens have been delivered as mRNAs at present. So what are the advantages of using mRNA as a vaccine? The data that we publish shows that it's much more potent than live virus vaccines, and it doesn't have any of the bad effects that live virus vaccines do. Live virus vaccines have safety issues. They're difficult to produce. The FDA hates them, while mRNAs, a single dose gives equal effects as a live virus vaccine and much better results. So this is a study delivering a PR8, which is an influenza HA, encoding mRNA. In the field of infectious diseases, it's believed that acute infection with the pathogen is the best immune response you can get. And in this case, you get an HA titer of about 1 to 500. A single injection of mRNA gave an HA a neutralization titer of 1 to 2,500. So 5 times better than acute infection with the pathogen. We recently published delivering a Zika encoding mRNA, both to BALC mice, where we had protection out to 20 weeks after a single immunization, or complete protection in rhesus macaques after a single immunization with Zika encoding mRNA. So to recap, what are the advantages of mRNA over recombinant protein are you don't have to go through the enormous process of producing and purifying therapeutic proteins. Every therapeutic protein, you have to reinvent the purification. So it's difficult, it's expensive. RNA is easy to make, it's a single reaction. You can also deliver intracellular proteins with mRNA. Comparing it to DNA and viral vectors, it's much safer. There's no chance of integration into the chromosomes. You get instant translation, and you can regulate how long the translation occurs, from anywhere to 2 weeks down to a couple of days. In thinking about what RNA can be used for, it's been recently described that RNA could keep us young by delivering rejuvenating factors, as well as a variety of other things. These are my collaborators. Katie Carrico is my chief collaborator in all of this work, and the funding sources. Thank you.
Thanks very much. Thanks very much, Drew. If right, there are going to be lots of questions, I'm sure. Please, could you use the microphone?
One question. Mechanistically, do you know how it's controlled in the cell? Do you think those modified mRNAs produced because they transcribed in the nucleus, where it might be different concentrations of nucleotides for synthesis? I'm thinking about the RNA viruses that normally replicate only in cytoplasm. So do you think mRNA that produced by viruses in the cytoplasm would have unmodified or modified nucleotides?
So it's actually both. And there are a number of viruses that have learned to overly modify their viral RNA. And in studies done 10 plus years ago, they actually went back and they removed the modification sites by changing the nucleotide sequence. They found no effect, but what they were doing was they were infecting cell lines in culture. And we know that cell lines don't really care much about modification. They never took those viruses with high or low levels of modification and put them in vivo to ask, did they have different levels of pathogenicity? And I think that's the critical experiment to do.
Hi. Years ago, some people published papers showing that in a stop codon, either UGA or UAG or UAA, in both yeast and mammalian cells, once the uridine is pseudorythylated by small nuclear RNA, then the stop codon can be recoded by certain tRNAs. So I'm wondering, in your modified mRNA, is the stop codon also modified? And if so, have you done a Western blot to see whether the proteins produced are actually a homogeneous or heterogeneous population?
Yeah.
Thank you.
So those studies were described in yeast and in rabbit reticulocyte lysate. We actually set up a very sensitive system where we put a firefly luciferase, a stop codon, and a Rna luciferase in a single coding sequence. And then we analyzed that with or without nucleoside modification. We saw no increase in read-through of the stop codon in mammalian cells. So we do not find that pseudouridinylation of a stop codon makes it leaky. In fact, we actually put IRESs in between those two sequences. And when you pseudouridinylate the RNA, there's less IRES activity, not more. So we saw no read-through of stop codons and no increased IRES, actually decreased IRES usage.
What is the half-life of your lipid nanoparticle system with a pseudouridine modified mRNA in the bloodstream?
So in the bloodstream, it has a relatively short half-life. I think it was best looked at with siRNAs, where the half-life was hours. You could still measure some LNPs in the circulation 4 to 6 hours after a single injection. We measure half-life by RNA function, by RNA translation. And we can see that with intradermal injection, translation continues for upwards of 2 weeks. We think that that's likely a depot effect, where the LNPs are sitting in the intradermal space and continually being taken up by incoming cells. So there, the half-life is very long. It's days.
Okay, last question.
All the immunoresponse studies, you guys use either lipo or lipo-based particles. Did you guys use anything else apart from lipo, like any polymer-based, like trans-IT or something?
So we actually searched about 40 different described compounds for complexing nucleic acids, including cationic lipids, cationic polymers, a variety of different LNP formulations, really every imaginable formulation we probably looked at. When we looked at them in vivo, the LNP deliveries were far superior, 10-fold better than any of the others. Most, but not all, did not introduce immunogenicity into the RNA. And we think it was probably the immunogenicity of the particle, not the RNA, that was being introduced. So most were non-immunogenic, some were.
Okay. Well, thanks again, Drew, for a great talk. Thank you.
Thank you, Steve, for this introduction. So I'm going to give a kind of review instead of my own data on what have been done in design and the delivery system for Messenger Rna. And of course, there are a lot of information that have been given by Drew and Louis, so it's going to be easy for me.
It's okay.
Okay,
You're not losing any time, don't worry. It's not started yet.
Okay, so from the empirical approach that has been done by Louis Pasteur for the first vaccination, nowadays we are now in this era of reverse vaccinology and the use of next generation technology that allows us to really design a very potent epitope for different types of organisms or for cancer cells. So in addition to the DNA or RNA, you can also use actually different types of new adjuvant, and I'm going to talk about that later on. So the goal of any vaccine candidates is to reach dendritic cells, which is actually the best player, And dendritic cells can activate naive T lymphocyte, and as a consequence, we get an effector cells, activation of different types of immune cells, as well as memory cells. So now we can really rely on DNA and RNA these last days. Okay, I'm going to switch this because I think that they have already talked about this. Dendritic cells is really, I'm going to comment briefly. It's a differentiated cell, so it's very difficult to transfect. So that's why mRNA is good. And as they have been told before, you can see that these dendritic cells, when it's mature, it's going to really interact with naive T cells through different interactions with co-stimulatory molecules. And then you see here a lot of cytokines that can actually activate Th1 lymphocyte, T lymphocyte, Th2, Th17, or Treg. What you have to keep in mind is also the fact that mature dendritic cells have a specific immunoproteasomes, which is called, proteasomes, which is an immunoproteasomes, because some of the subunits are replaced by a specific one, LMP7, LMP2, and MECL1. And actually, the epitopes that are processed by these immunoproteasomes are quite different than the one, for example, that is expressed on melanoma cells if you are talking about MHC class 1 presentation. So it's really important to know how they are acting, so you can knock down, for example, these different types of inducible subunit, so you can have only these constitutive proteasomes working, and then you can match the two types of epitopes. Another key issue for vaccination is the presentations of antigen. So there are presentations through MHC class 1, which is actually all of the genes that are expressed inside the cells. They go through through these proteasomes, and then it can be expressed here via VCMH1 and through the ER targeting. The second one is the CMH2 presentation, class 2 presentation. So here, proteins or microorganisms from the outside will enter via endocytosis, reach what is called the C2V compartment, and then you can have here there is a charging of VCMH2 class, VCMH2, and then you have a presentation through VCMH2. But recently it is known that actually the antigen from the inside of a cell can also go to the C2V, and then you can have a presentation through the CMH2 as well, so it is called a cross-presentation. So again here, you can play by using a LAMP1 targeting motif, which actually allows the proteins to go to the C2V, and then the proteins, the antigenic protein, can be charged on the CMH2 as well. So this is two types of tricks that you can use if you would like to have a both CMH1 and CMH2 presentation. So I think that this is the plain motif of mRNA. You have this cap structure, and then you have this poly-A. And here is the open reading frame. this cap is really important because it's really playing a role for the stability of a messenger RNA. There are different types of cap analog that have been done because if you are doing in vitro transcription, unfortunately, half of your molecules will have a reverse orientation. So by using this RCA or this beta S RCA, you force the system to go in a good orientation, so most of your molecules will be good for translation. There are also different length of poly-A that you have to use to stabilize our messenger RNA. I just have to say that actually there is a loop between the cap and the poly-A during the first step of a translation. So I think Drew has actually given a very nice introductions of the sensing molecules that are mechanisms that are inside the cells. You have plenty of them. And of course, messenger RNA, exogenous messenger RNA can be sensed by those sensors. And in the end, you can have these reductions of a translation. So, but this immune stimulating property is really important because it can be of benefit for immunotherapy. So we need to get a good balance between the good translations and reductions of immune response. And actually, it has been told already by previous speaker that the molecular structure of this delivery system can have an effect on this immunogenicity. Today, there are two forms of messenger RNA. The one is the usual one. You can get it, both of them can be obtained through in vitro transcription, but this one is not an amplifying one. And the second one is a replicon RNA or self-amplifying RNA. So how it looks like? So you can get it from genomes of alpha virus and flavivirus, and you can see here these are 5' with cap, and here you have a different in alpha virus replicon you have a non-structural proteins 1 to 4, and for the flavivirus we have this NS1 to NS5. Here there is a replicase that can be expressed, and then you will see once it's delivered inside the cells, you will have actually you will have this transcription of translations of a replicase, and then you will have a negative strand of your RNA, and then it can be auto-amplified by replication in positive strands. I just want to show for the last minute, we'll focus on how you can actually modify the immune systems to improve the efficacy. The first one is a trimix technology. Trimix technology has been proposed by Chris Tillmans in Therapeutics in Brussels. And you can see here, it's actually based on three points. The first one is CD40 ligation on DC to get an activated CD4 plus T cells. And the second one is to mimic a LPS binding on a DC, which actually allows to have a very active form of TLR4. And the last one is a ligand, is an interaction between a CD70 ligand and CD27, which is expressed in activated B cells, naive T cells. So by expressing via mRNA these three molecules, you can actually have a very good immune response. Because by having a very good DC via CTLR4, you reduce the productions of this Treg. You have activations of naive CD8 cells, and also a lot of cytokines that can help for the productions of Th1 phenotype. So the trimix technology has been used to mRNA plus mRNA encoding TAA antigen, which is tumor-associated antigen. It's an intranodal delivery, and you can see that there is actually a positive activations of a DC cells through the expression of these co-stimulatory molecules. And you can see here that mice that has been treated with this intranodal delivery vaccination, there is really an eradication of tumor growth and an improvement of the survival. The second part is on my talk is on delivery systems. Of course, one of the problems with this intranodal delivery that have been done, or the DC pulse mRNA, is that the mRNA is very fragile, so as shown before, you can have a very low efficiency in terms of translation. So there are a lot of chemical and physical delivery that have been tested for transfection of messenger RNA. So the Lipo Plexus, LNP, and also the Cationic nanoemulsion. Two types of physical delivery, electroporation and sonoporation. I'm not going to talk about this because I don't have enough time, but anyway, anyhow, all of these different types of chemical delivery, and it's the same for the physical one, you need to go inside the cytosols to get to be reached by the translation machinery. So, of course, most of these polymer and lipids, they are using a proton sponge effect or destabilizations of the endosomal membrane. The second part is an RNA-active technology, which is done by CureVac. It's a CureVac technology. Here, they are used a protamine, which is a self-condensed, which is a which replaces histone in sperm, so condensing nucleic acid. It has been shown that it induces innate immune stimulation through TLR7 activation, so it's very good for vaccination. Okay, so CureVac actually made a composition of RNA they called RNA active, 50% of free RNA, and 50% of mRNA complex with protamine. These 50% of RNA are actually modified to be stable, and it's actually phosphorothioate modification. In mice, they found that there is a boostable and balanced humoral as well as T-cell mediated antigen-specific immunity, and the productions of memory T-cells, which is really one of the goals if you would like to do vaccination. So this is an example for in mice with EG7 OVA model. So you can see here that compared to ovalbumin mRNA peptide, ovalbumin peptide, sorry, and synthecal peptide, here we really have a very high decrease of gross tumor thanks to the vaccination made by just an ID injection with 10 microgram of this RNA active encoding OVA. So it's 2 on day 1 and after 3 weeks. And of course, you have a very good survival here. They have used it for clinical trials. The prostate one CV9103, which is a cocktail of different types of antigens, prostate cancer. Unfortunately, there is a failure, so it has been stopped. But another one is still on, NSCLC, encoding different types of antigen, MHC1, MHC2, and ISO, surviving, and 5T. The second delivery system that I would like to talk about is the Lipo Plexus. So again, if you look at it, very simple type of lipids, and you see you have a low dose, I would say, and the different types of administration have been tested. So I just want to point at this one, but Ugur Sahin will explain a little bit more on Friday, I guess, or Saturday, I don't know. So here, what they have done, they actually trying to fine-tuning the ratio between the two lipids, DOTAP, DOTMA, DOTAP dope, DOTMA cholesterol, and DOTMA dope, and they found that in a range of negatively charged or nearly neutral of this lipid ratio, they can have a splenic targeting. And I think Luis has presented a few of it because you are working with, I don't know. So you can see here, it's a splenic targeting, and if you use a mice that you eradicate all of the dendritic cells, there is no more luminescence inside the spleen when you are using messenger RNA coding for luciferase. And here they detected that DC, plasmacytoid DC, macrophage, and NK can be actually transfected inside the spleen, but mostly the DC. Here is just an illustration of one of the in vivo experiment. So it's a melanoma mimicking metastasis, so through IV injection. So it's a therapeutic vaccination. So they inject TRP1 lipoplex mRNA and for 1, 2, 3, 4 times. And you can see that the growth of a tumor is really delayed and is very low compared to the placebo or the control one. So see, this is a number of metastases, it's really cleared off. Another very widely used delivery system is the RNA lipopolyplexes, and we are using this as well. So it's a combination of cationic polymers to compact the RNA, and then you add the liposomes, and then you can have an encapsulation of your polyplexes inside the liposomes. So see, again, it's a low amount, I would say 30, 12.5, or even 7, 10 microgram. And you have a positive charge, in our case, 150, 100 to 150, and we have made a targeting on this. So I just want to take the opportunity to show you an example of the targeting of the actually the protein coded by the mRNA to the C2V, where you can have a cross presentation. So here we have an ARCA as a cap of our messenger RNA, the MART1 sequence on which we put a LAMP1 delivery signal. And you can see here the tumor volume. If you add the two types of the RNA, you can really have an increase, a beneficial synergy effect compared to the MART1 only or the MART1 LAMP1. Concerning the lipid nanoparticles, I'm not going to talk more about this because I think Luis Andrew has given a lot of details, but it's really fascinating and giving a very good result. So you can have a polymer and you can have different types of lipids. And I'm going to switch this because it's exactly the same as he presented. You can have a neutralizing antibodies. You can have a very good inhibitions of infection as well. The last one is cationic nanoemulsion. So the cationic nanoemulsion has been used for replicon RNA, which is very good. So the replicon RNA is actually more than 10 kilobases of a messenger RNA. Very tough to be used because it's very long, it has a lot of secondary structure. And therefore, you can have a very intrinsicate interaction. So the problem is more interactions you have, you need to have a decomplexation inside the cytosol. So very difficult to actually to fine-tune between the complexation and decomplexation of these very big molecules. And of course, the bigger are the molecules, the increase, the signaling by the sensor are also big, and you can have a decrease of the translation. So this cationic nanoemulsion, it's composed of twin AT with an oil phase containing span, 85, DOTAP, and squalene. So you have something like 86 nanometer. Actually, I couldn't find any zeta potential of this in the paper, so maybe, I don't know, I couldn't find it. But still, you have a similar dose as messenger RNA here. And Brito, I think it's, Luis has used it already, so he can answer the questions if you want. So here, you can see that two immunization intramuscular, day 0 and day 21, you collect the sera and you look. This is a RSV, the glycoprotein F encoding mRNA. So you have a self-amplifying or just the mRNA non-amplifying. So you can clearly see here that the level of the specific IgG obtained after the vaccination is mostly close, very close to the VRP. The second, you see that 15 microgram, and you can even get it for, this is for just in PBS, and if you put it in CNE, you see that you have a very high expressions, productions of IgE. And see the pDNA is actually very low, is low. This one, I picked the data for immunogenicity in macaque, and again, you can see that there is also a high expressions of IgG. And this is obtained after intramuscular immunization on day 0 and day 28 with 78 microgram, which is not too big. But, okay, I told you that by just fine-tuning the different types of lipids or different ratio of your delivery system, you can get a very nice targeting. Still, I think that you can improve this targeting by using ligands. So here you can see the DC has a specific ligand, has a specific receptor that you can actually target. Mannose receptor, DC sign, TEC205, TECTIN, TECTIN1, TECTIN2, and Langerin. So you see, these two, they have a mannose motif. And all of this one has a beta-glucan, but most of them has a mannose motif. So we have used our LPR and target them with a first targeting with just one mannose motif. And by doing this, we found that after IV injections of our LPR nanoparticle, mRNA nanoparticle, we can actually improve the dendritic cells that are transfected in the spleen from 2.5 to 13%. And what is also good is that you don't have a very good difference in terms of physicochemical characterizations of a nanoparticle. But if you are looking at the literature of this mannose receptor, you will see that actually they don't have just one mannose motif, but they have a very variety of range of number of mannose motif. So we started to do this one, to build this one, to produce this one. This is a 3 mannose-dilated to improve the avidity and see if it can have something good. So first things we have done was to see if the targeting is really okay. So we used here HEC293 stably expressing DC sign. This one is a langerine and this one is DC. So I think you see the black compared to the other, that there is a very high improvement of the uptake of those LPR nanoparticles inside these different types of cells. We use it to vaccinate mice through ID vaccinations, 1' and 2 boost with E7 mRNA. And this is the yellow spot. So you can see that using the tri-mannose-dilated LPR, we have really improved your vaccination. And of course, as a consequence, there is a very good survival if you compare it, this one, to the mono-mannose, and of course to this one, it is just the mannose LPR without RNA. The last slide I would like to share with you is the type 1 interferon story. Type 1 interferon is one of the cytokine that is expressed after if you are actually activating DC. It's very tricky because depending on the conditions and depending on the way you are using it, you can have whether a very good stimulation or a T-cell apoptosis. So what happens? Type 1 interferon, you can have actually two, you need to, 2 signals are required here for these activations. So if your first signal is the TCR, I mean the docking with a TCR, receptor T here, and after that, the TCR is going to activate the signaling inside the cells, interferon, type 1 interferon is going to be produced, received by the IFNA, which is the receptor of interferon. Through this way, we will have an increase of STAT3 transcription factor. And then you have a very good immunization because you have a T-cell proliferation and T-cell differentiation. If you are unlucky, you will have first the productions of type 1 interferon. And this can happen if actually your delivery systems is prone to activate this. And by having this, you will get a second signal through the TCR, and unfortunately, you will have a T-cell apoptosis. So I think that if you want to make a delivery system for messenger RNA in terms of vaccination, you need to check all of these different types of cytokines to make sure that actually you are in a good way. So just to summarize, all of these talks actually I've told you, I hope, that mRNA is really a versatile platform for antigen production. And you can use polycistronic messenger RNA, which is different open reading frame sliced by IRES or P2A, or a self-amplifying mRNA to have a long-lasting expressions of your protein. The trimix technology can actually be added with this delivery system, and then you can boost the expressions of your antigenic proteins and your immune stimulation. And of course, even though the first of clinical trial of the RNA active is not has failed, I think that they're still very, very, very nice, because it shows that they are safe, and there is no real toxicity problem. I'm sure that you have seen that this fine-tuning of the different types of your delivery system can really, at the first step, allows you to go to a target tissue, and then you can add a more selective ligand to improve the targeting. So there are still open questions. How could we improve the translations and the antigen processing? And I think here we need to go deeper in the translation mechanism, understand these very tough helicases involved, get a clear combinations of immune response for protection or therapeutic treatment. Maybe as function of delivery systems, you can have a better use for, use it much more better for protection or for therapy. And then the last one is very few actually papers are reporting the delivery systems as function of mRNA size and structure, and what type of sensor are activated. And of course, you can use a messenger RNA vaccine with other therapy. I would like to thank my collaborators, and thank you very much for your attention.
Thanks very much. If anyone has questions, please come to the microphone. While you're thinking, I have a quick question on the production of mRNA. So you talked about two methods. One, the in vitro transcribed, and the other synthetic. And I think, so does synthetic mean extracted from virus?
I put a slash. Synthetic means it's also in vitro transcribed, because for actually the mRNA people are using synthetic or in vitro transcribed. That's why I put the two of them. Okay. Yeah, it's exactly the same.
So it made me think, is there a chemical synthetic?
Yeah, I mean, the chemical is actually you add your modified nucleoside to improve the stability. This is just the thing you add. Because when you do your productions, you have your plasmid, in which you have your genes of interest, promoter of T7 or sp6 polymerase, okay? And then you can have a transcription in vitro, and during this transcription you can add the pseudouridine, for example, or the different types of modified nucleosides, so you can have a synthetic messenger RNA, synthetic and messenger RNA.
Okay. Kevin.
Very nice, comprehensive presentation. One of the areas you touched upon, which I think is important, that the field will struggle with probably, is persistence of expression from mRNA.
Yeah.
And most of it's 2 or 3 days expression, especially when IV dosed. If you IM dose it or IM electroporate it, it's a little bit different story. But you were proposing a replicon approach, I believe, or you talked about it. But the problem with those constructs is they become huge.
Yeah, it's working.
You take an RNA that's normally 2,000 bases,
I know.
and one that's like 10,000 bases
I know.
to get all of that stuff in there. So is there any other solutions that you know of to create persistence without resorting to that replicon system?
I don't know, maybe you can help me, but there are a lot of, there are some indications that depending on the locations of your messenger RNA inside the cells, you can improve the persistence, okay? It's called the zip code. But, I mean, no one actually can really solve the problem. It has been shown in Drosophila, for sure, but in eukaryotic cells, we don't know. So what we are trying to do in the lab actually is to do an intracellular, routing of the messenger RNA once it's delivered, just to really to understand where it's go, because we have different types of delivery systems, and we know that some of them have a very high copies of proteins, long-lasting, and the other is very short one. So clearly, in my opinion, intracellular trafficking could really help. But here, you need to detect a non-modified RNA, messenger RNA. Otherwise, can be a mess. I don't know. What do you think, Joe?
Okay, I think we can perhaps discuss that further over coffee. So I'd just like to thank Chantal again. And of course,